All right. Thank you so much for giving me this wonderful opportunity. It's a real privilege to be here at the Ethical Society and to see so many people. In fact, I have never had so many people attend one of my talks. So this is, this is amazing. So I'm honored, and I'm really delighted. So thank you. So first of all, I want to start by thanking the Ethical Society for inviting me to come here and say a few words about this topic. And this is not what I've studied. This is not my area of research. But this is something I teach students, both in my undergraduate cell biology classes, but also in graduate courses in developmental genetics. So I have been doing these classes for many years. So I've built up some ideas and material that I thought you might find interesting. And more importantly, I have the University of Missouri system has this program called Presidential Engagement Fellows, where they like faculty and staff to go out and share their knowledge with the community and engage with the community. And so I again thank the program for giving me this chance. So as an outline, I'd like to tell you a little bit about what are stem cells and how stem cells are made and some ethical issues associated with that. And finally, how stem cells are being used and how they can be used, and some of the ethical issues, again, associated with that. So to start off, I know that this is a very diverse audience, and I'm sure there are people here who could be medical doctors or in the humanities who have engaged in these topics over time. So I want to start with a disclaimer that I will identify some ethical concerns, but I don't have any answers for you. And surely you will have a Q&A session, so I'll be happy to take your questions, but I'm approaching this from the scientific side, but I'll be happy to try and answer any questions you have. I'm not an expert in either stem cells or in ethics, okay? But I did get a good night's sleep last night at Home 2 Suites. So hopefully I'll do a decent job, so let's see what happens. So what are stem cells? So if you think about your own body, and you think of all the different tissues you have lung cells, you have pancreatic cells, heart muscle, red blood cells, skin, neurons. These are all different types of cells in your body. They all are uniquely equipped to do different functions. Right? But when you think about so they are considered what are called differentiated cells. They are highly specialized to do different functions. But they all started out, the very beginning, from that initial fertilized zygote, a single cell. And over the course of development of the human embryo, they diverged in their potential, and they ended up acquiring these very unique and singular properties or functions. You could think of them as maybe unipotent cells. They're doing one thing, and they do that one thing very well. So how did it happen? So one way to think about it is the nucleus, which is the storehouse of the DNA, the genetic information within that cell. It has been somehow programmed to do one thing. So the nucleus within a lung cell is programmed to do one thing. The nucleus in your skin cells are programmed to do one thing, and they do this very well. And so the egg cell had the ability to do hundreds of different things. But then those nuclei progressively got programmed to do restricted things. So when you think of that egg cell, the fertilized egg, the oocyte, the egg cell is fertilized by a sperm, that is considered totipotent. It has the ability to become anything it wants. Same thing with a couple of cell divisions later. If you think of the morular stage, where you have a small collection of cells, they are again considered totipotent. They can do anything they want. They have endless potential, limitless potential to form all the different types of cells in the body. Right? So you could think of either the fertilized egg or these morular cells as the original stem cell. They have limitless potential. From that stem, you can get different types of cells forming that you can then see in the different organs and different tissues in your body. So then the question is, well, are there any stem cells in adult tissues? Are there any stem cells in your lungs, in your heart, in your skin? And it turns out that most tissues have some resident stem cells. So if you think of the gut that's lining your small intestine, those cells are sloughed off all the time. Your skin is getting sloughed off, okay, due to wear and tear, and there are new cells that are coming up from underneath and replacing those. So many, many different types of tissues in your body, they get replenished over a period of time. Some of them get replenished every few days, others few weeks, others months, and some, they never get replenished. For example, I've heard of this. They say that the nervous system never undergoes regeneration because you don't have stem cells. For the most part, that is correct. There are some exceptions. And same thing, the germ cells. So a woman has a certain number of oocytes, and beyond that, you don't form any more. And so there are some tissues where you don't get stem cells to form more of those types of cells. But you know examples, again, from our own body, the bone marrow cells. They are a rich source of stem cells, and they are used now, for example, for cancer patients, right? You can do bone marrow transplantation for people with certain kind of cancers, and then you can treat them and then repopulate with stem cells from other healthy donors, and they can have healthy blood. So there are already examples of adult stem cells that have been used and that are very important for in medicine. But the challenge with all of these cells, so again, this is just showing different types of stem cells that come out of the bone marrow. For example, all of your red blood cells, white blood cells, they're all being formed every few weeks, and they're coming up. They are being recycled, and they're coming up once again. But these are very difficult to study, right? So they are very important, and there are restrictions and limits to how we can either collect them, isolate them, study them, and maybe use them to help benefit more patients. So that is always challenging to study these adult stem cells. So the question always was, is there a way to make stem cells in quantity? Can you make them in large amounts? And that was the next challenge that scientists want to tackle. Can we get large quantities of stem cells to study? So before I can answer that question, I have to kind of tell you a short story. Not about home to sweets, but how people have been thinking about these stem cell questions for a long time. So I told you about how the original egg, the nucleus within that egg, has the ability to form many different types of cells, but then it progressively gets reprogrammed to do only certain things. Totipotent can do a lot of things. Unipotent or multipotent can do fewer things. So this question was first asked in the 50s by a scientist named John Gurdon and his colleagues, and they asked, what if you take a differentiated cell, like a skin cell, or a nerve cell, or an epithelial cell, in this case from a frog and from a tadpole or from an amphibian, and what kind of ability does it have? Can you somehow reprogram it to form all the cells in an embryo? So how do you reprogram a differentiated cell's nucleus? And he discovered that you could transfer that nucleus into an egg cell, and you can reprogram it. And that's what is shown out here. You take an intestine, you isolate all the epithelial cells, you take the nucleus out, and you put it within a recipient egg. This is again a frog egg. You take its own nucleus out. So this is now the nucleus from the skin cell. And this will now develop into an adult frog. And you know that the tissues in this adult frog were derived from the cells, the nucleus that came from these cells, because you can have them in different colors. This frog would be an albino frog, and the egg comes from a pigmented frog. And so that is this pigmented frog. That's the one donating the eggs. The nucleus, when it then forms an adult frog, they all give rise to albino offspring. So it tells you that the nucleus from that skin cell was reprogrammed to form adult frog cells, but you know that they all came from the donor cells and not some kind of accidental case where you didn't do something to the nucleus, didn't work properly. So these are your stem cells. All of these cells have the ability, and those are the stem cells that got reprogrammed. You got it by reprogramming this nucleus that came out of these differentiated cells. Okay. But this is an opportunity now to study stem cells in quantity, but this is a vertebrate, but it's not a mammal. So then the scientists asked, can we do something similar in mammals? And sure enough, people discovered in the 80s and 90s, you could do this in mammals, where you could take cells from differentiated cells in, for example, cattle. You can take the nuclei, you can put it inside an egg from another cow, and then you can get animals that look like the donor. Okay? And again, you have these stem cells. You have the stem cells that were derived from the nucleus that got reprogrammed from your donor cells. Okay? And this you might know in the 80s and 90s, many of you may remember cloning of Dolly the sheep, Snuppy the dog in Korea, and of course at Mizzou, they cloned a lot of green pigs. It seemed like a novelty then, but it turns out to be very important because you may have heard of the pig heart transplanted recently, about a few weeks ago, months ago, and the original work for generating that particular type of pig so that you don't get immune rejection was due to technology that came out of the ones that made the green pigs at Mizzou. So stem cells can be isolated and used for research. So here are the stem cells. These are now from mammals. You can take these stem cells out. This is called the inner cell mass. You can take it out, you can study it, and you can use it for research or therapy. So this sounds great, but now come the ethical concerns, right? So you have two things here. Those stem cells are very useful, and they can potentially give you life-saving cures for people who need them, whether it's in neurodegenerative diseases. Okay? Those can be useful. You can do cell transplantations and help people maybe with Parkinson's. But on the other hand, in order to get those stem cells, you have to destroy the embryo. Okay? You are destroying, in this case, if it were for human applications, human embryos. So this was a very big ethical concern, and it's natural. Which one do you choose? So you have to destroy the embryos. You have no other way of getting at those stem cells. So people said, well, why don't we make stem cell lines? We will use a few embryos, we'll make a few stem cell lines, and once you make those stem cell lines, they'll be useful. They had some utility, but they were not perfect. They didn't really always work out. And the other problem again is, if you do want these stem cells, and you are willing and prepared to destroy embryos to get them, but there's still only a very limited supply of human embryos. And the only way you get them is by doing IVF. People do IVF, and once you select one or two good, healthy embryos for implantation, the others are discarded. They cannot be used for any other purpose, but there are, again, ethical issues associated with that. So it's not a really easy answer, well, why don't we use it for something that will benefit others, right? So there were all these challenges, and Scientists were thinking, is there some other way we can make stem cells, okay, without destroying human embryos? And that leads us to this idea of programming the nucleus. In the case of programming here, the programming was done by transferring the nucleus into an egg-like environment, into a naive environment. So something in the environment was reprogramming the differentiated nucleus to revert to a very primitive state. So scientists asked, is there some other way of programming the nucleus to revert back to this primitive state? And a group of scientists in Japan, led by Shinya Yamanaka, he did some of this work in San Francisco, but his group was able to do this reprogramming both with mouse. You can take a differentiated cell from a mouse or from a human, and you can reprogram it to a stem cell by using what are called defined factors. And what we know by defined factors is you express a small group of genes in these cells, and you can convert them into a stem cell. And these were called induced pluripotent stem cells, or iPS cells. So essentially, you're taking a differentiated cell, like a skin cell or a fibroblast cell, which is found in your connective tissue. You express different genes in it, and over a period of time, you make it into an iPS cell, and this iPS cell now has amazing abilities to form any different type of cell you might be interested in. You can induce that cell to go in any one of these pathways. Okay? This was a tremendous advance, and within a few years of this discovery, both John Gurdon, that first scientist was working with those frog eggs and finding out what potential they had, and Shinya Yamanaka, they were awarded the Nobel Prize for providing this amazing insight into how you can convert a differentiated cell or program a nucleus into something that is more stem cell-like. Okay. So we have come a long ways. We now have the ability to make stem cells without destroying embryos. Okay. So how can they be used? Or how are they being used? Okay. So again, this is something, I'm going to have three different parts to this. One of them is something I'm going to call studying disease in a dish. Okay, and this leads to something that you might have heard in the popular press, something called personalized medicine. Second one is growing tissues and organs, transplantation, and treating infertility. So studying disease in a dish. So nowadays, for the most part, if you had a certain disease, there are drugs available, but they are almost always one size fits all. Okay? There are clinical trials that are done at certain doses, certain regimen, and then the treatment is done. But this treatment doesn't always work, whether it's for cancer or for some other disease, it doesn't always work. If you use the same treatment for the entire population, some people you may see a beneficial effect. In others, there may be no effect of the treatment. In some others, there could be actually an adverse effect. Things are not so good. So how do we overcome this? Because there is heterogeneity among us. We don't all respond to medicines in the same way. So then this concept of personalized medicine has come about in the last 10 years, where you can imagine patient-specific treatments, okay? Using a variety of different methods. If you can make a treatment and a drug that can work differently for different groups of people, you may end up getting a beneficial effect in all of those different groups. But then the treatments have to be engineered or designed for everyone in a different way. So how do you do that? How do you discover patient-specific drugs or treatments? And this is where stem cells are going to prove to be very important, because you can now make stem cells from each and every one of us. You can take a skin biopsy. You can take those cells, put them in culture, treat them with these factors, and you can make stem cells that can then become any type of cell in the body. So if you had a heart condition and you wanted to find out, and it can be expressed, you make stem cell, make it become the heart muscle, maybe they will express the defect that's seen in your intact heart. You can then treat it with different chemicals, and voila, maybe you'll be able to find a compound that works specifically in your case that may not work for somebody else. And that is what is illustrated out here. For every person, you take a biopsy, you make patient-specific stem cells. You can then take those, in this case, this example, it's a nerve cell. Those nerve cells from this patient are defective. You find a drug that works for that person, makes those nerve cells healthy, and therefore now you can treat that person with that drug that is having this very specific effect in culture or in a dish. So you're trying to model the disease that this patient has in a petri dish. Alternatively, you can take those stem cells, you can correct whatever genetic defect is there in vitro using, you may have heard this word CRISPR, some of you may have heard it, you can modify the DNA, you can edit the DNA, you can change the defect in the gene, you can then make those neurons once again. This could be in Parkinson's, perhaps. You can then put it back in the person and maybe restore some of the function that they are missing. So it's going to turn out that patient-derived iPS cells could represent one of the best model systems. I haven't told you what a model system is for understanding and treating and curing diseases. Okay. Do you have another 10 minutes? 5 minutes? Okay. So I'm going to then skip this. So, of course, now we can use, there are many clinical trials already underway to be able to use stem cells to correct different types of diseases and metabolic disorders. But there are concerns with stem cell, iPS cells. There are issues with tumorigenicity and immunogenicity. The other way of looking at this is tissues and organs, growing them. So those iPS cells that are made, they can act very much like the cells in your body. For example, here is an intact mouse embryo that has been stained for 2 different tissues very early on in development. This is about a 5-day-old mouse embryo. Here is a mouse embryo that was constructed by stem cells in a dish. You won't be able to tell them apart. Okay. So those stem cells are able to behave just like the mouse embryo cells are behaving inside an intact mouse. Okay. This is just showing you stem cells that people put in culture and let them become different, what are called organoids. They can form eyes, they can form lens. They don't look perfect, but these are early days. So soon we may be able to form many of the different cell types that maybe are affected in different kinds of visual eye disorders and maybe help patients out that way. And in this case, we are using their own tissue. So if you wanted to transplant it back into a person, you're not going to have any immune issues because it is going to be self. It's going to be your own cells that were converted and placed back inside you. This is showing you something called a brain organoid. You can form bits and pieces of the brain. And they look remarkably like brain tissue that forms inside an embryo. So one other way of looking at this is you can even make what are called human interspecific chimeras. You can take human stem cells, you can implant it and put it inside the inner cell mass in a pig embryo, you can get human-pig chimeras. Okay. What this then allows you to do is to grow human organs in a pig. So you could grow a human heart. This is a little bit different from the pig transplant that was done. That was a pig heart put in a human. But that pig tissue had been modified so it would not cause an immune reaction. But you could potentially make a human heart in a pig. And it's not doable now, but it could potentially be doable using these stem cells. Okay. And you could edit those stem cells and correct whatever genetic defect is there and put an organ that is now completely normal. Of course, this then raises the issue of purely growing animals for just harvesting their tissue. Of course, a lot of us eat animals now, so, but this is a little bit different. Is it okay to just grow some animals just so you want their heart or the lung that's growing in it that belongs to you? I will stop with this. I did have a thing on animal research, but I think this is pretty cool. So you can use stem cells to make sperm and egg. Okay, think about it. You can make, so these are sperm-like cells, and these sperm were used to fertilize mouse eggs, and you can get perfectly healthy mouse babies. Or you can take stem cells, convert them into egg cells, oocytes, fertilize them with normal sperm, and you can get very healthy mouse babies. So you can get huge amounts of eggs and sperm from stem cells. And those eggs and sperm are perfectly functional, perfectly viable. So this can be cure for infertility. I mean, people have all kinds of reasons why maybe they are not able to have children. So this could be a way of potentially curing infertility, because you can take someone's tissue, convert it into stem cells, make eggs. But some of the issues with this are you do get a large number of IVF embryos, because you're going to then get those eggs, hundreds of them, and then you have to decide which ones you're going to implant. What do you do with the rest? But this last thing is really, really weird. I could take a biopsy of my thing. I could make iPS stem cells from my own tissue, but that can be made into either eggs or sperm, or both. And I could take my own stem cell derived egg and my own stem cell derived sperm and make a new animal, a new embryo. What is that? It's not doable now. It's on the way. This is all within the last few years. So the technology is there, it can happen. And so these are some things for us to consider. Okay. So I'm going to stop there because I don't have time to talk about ethics of animal experiments. But what I will say is that animals have been used for a long time for conducting experiments because it was easy to do. No one cared until the 50s whether it was okay to do animal research. And that's when people said, no, we have to be really careful. And there were what were called the 3 Rs: reduce using animals, refine the methods, or replace animal use altogether. And I think that stem cells, having stem cells, is really going to represent one of the best ways of not having to use animals in research. If you can model a disease in a petri dish, you can avoid having to use animals. So with that, I will stop, and thank you very much for your attention.
Thank you, Dr. Chandrasekhar.